Hi everyone. I'm continuing on section 6.2, environmental conditions that influence microbial growth. And just wanted to talk about a couple more environmental conditions. One is pH. And we talked a little bit about pH already. We said pH 7 is around neutral. Human blood is 7.35 to 7.45. So we function at fairly neutral pHs. And so do very many microorganisms. They say they like the range between 6 and 8, and strong acids and bases can be very damaging. You can imagine that our normal microbiota and any organisms that cause disease related to humans or most animals will survive also, will prefer these neutral pHs. The ones that are kind of the exception are these organisms we call obligate acidophiles. And these are organisms that have evolved in acidic environments, and their metabolism and protein structure is adapted to these acidic environments. And so here's a couple examples. Euglena, which is an alga, grows in acid pools between 0 and 1 pH. That's really low. There's an archaea that lives in hot coal piles. I don't know. I know there's a lot of sulfuric acid associated with coal. And so they live in a pH 1 to 2. So these would definitely be acidophiles. And because they're so well adapted to their low pH environment, they'll actually die at pH 7. We also have acidophiles that are common in our day-to-day environment. You know, some of the microbes we use to make things like pickles and other fermented foods like sauerkraut are acidophiles. And even we have some somewhat acidophilic organisms that live in the human vagina because they produce some acids and discourage the growth of other microorganisms. Molds and yeasts in general are more acid tolerant. So even just everyday mold and yeast, they're a little bit more tolerant to acid than most bacteria. And so they would be more likely to grow on foods that are somewhat acidic, like pickles if they are spoiled. Another environmental condition to consider is osmotic pressure. Now, we talked about osmosis, and we said that most living organisms can live in a hypotonic or an isotonic environment, especially, well, if you have a cell wall, most microorganisms can live in a hypotonic or isotonic environment. But there are organisms that can live in hypertonic environments. There's always an exception in the microbial world. And so these microorganisms that like these high concentrations of salt in particular are called halophiles. There's some examples here of some halophiles that live in salt lakes, ponds, and other hypersaline environments. There are, like the Dead Sea, I think is maybe 25 or 30% salt. Now compare that to the ocean. Ocean water is about 3% salt. So it's a great big difference compared to the Dead Sea. The Dead Sea is located between Israel and Jordan. And you'll see vacation pictures of people in the Dead Sea kind of floating in the water reading a magazine or reading a book because it's so dense with all that salt in there that you can float really well. And the Dead Sea was called the Dead Sea because they thought nothing lived there. It's dead. But it turned out there are halophilic microorganisms. And so some of these are only found in areas such as that are super salty. So again, these might be organisms that you may never come across in your whole life unless you go to a special place where they evolved. Another group, facultative halophiles. Remember when we said with oxygen, a facultative anaerobe means it can grow anaerobically, but it doesn't have to. Same thing with facultative halophiles. It can grow in a high salt environment, but it doesn't have to. They aren't typically quite as resistant to salt as the obligate halophiles, but they can survive in high salt environments. And even our just everyday staphylococcus that's found on our skin can grow in salt that's up to 20%, which is pretty high. And the reason is because our skin is very dry, and it therefore the salt concentration relative to the bacteria is going to be high. So a lot of skin bacteria can be considered facultative halophiles. So these are not uncommon. The last thing I wanted to talk about is just to revisit that concept of biofilms. We talked a little bit about biofilms when we talked about extracellular polysaccharides and surface layers that some bacteria make. And we said that these slime layers that some bacteria make allow bacteria to group together in biofilms and attach to surfaces. And the reason why I'm talking about this in the growth chapter now is that we kind of have always thought of bacteria, especially doing research in the lab. We put bacteria in some kind of broth, some liquid, and we let it grow up and they float around and we look at them under the microscope. We think about infections that way too, the bacteria kind of floating around, maybe in the middle of the ear when you have an ear infection, there's bacteria floating around. And what we have found out that in nature, bacteria are much less likely to be floating. We call them planktonic. If there's liquid and a surface, you'll have bacteria that are floating around that are planktonic. But a lot of bacteria will stick to surfaces and form these stationary communities called biofilms. And so here it says biofilms are communities of bacteria and other microbes that are attached to a surface and to each other, forming a multi-layer conglomerate of cells and intracellular matrix, that slime layer. That's all that slime. One of the surprising things is even in infections, like middle ear infections, instead of the bacteria floating around in fluid, it's actually they're attached with these slime layers to the surfaces of the middle ear. And biofilms are really important. We're going to talk about them again when we get to controlling infection and antibiotics. But this is a really good little video. You can watch that one. And the same thing, they have similar thing right here, but I kind of like the video. I thought it was a little better visual. But what happens when you're making a biofilm is as long as there's a surface and some food and some moisture, and obviously the right pH and temperature, bacteria, if there's bacteria around, they're going to stick to the surface. Oh, here it says, here's the gauze fiber. And this orange is the green's the gauze. The orange is the extracellular matrix, the slime layer, and the blue are the bacteria. So on the gauze, the bacteria stick. And so you're going to have a surface, bacteria are going to stick to that surface. And those bacteria are going to start making that slime layer. And that's going to allow them to really attach to the surface. And then they're going to, other species can join in. So if this is something in your mouth or something in the environment where there's other kinds of microbes around, you're going to end up with a mixed community of microorganisms. And they're all going to kind of trap into this slime layer. And it's going to be kind of interesting because the conditions up here in the slime layer, in this whole biofilm, are going to be different than down here. For example, there might be more oxygen up here. So the microbes that grow in the different areas of the biofilm can also be different. One of the things we know about biofilms is that microbes can dislodge from the biofilm and they can move to other parts in the body. So you hear about things like concerns that when you do dental work on the mouth of somebody who has gum disease, so they have pathogenic bacteria, that those bacteria might get into the blood and travel to another part of the body like the heart. And so this is one really common way that infections move from one part of the body to another, with little chunks dropping off of biofilms. So biofilms are very common in the human body, they're very common in nature, and they're a very common way that bacteria grow.